Good morning, everyone. It's great pleasure to be here talking to you all. My name is Saeed. I'm a senior research fellow at the Francis Crick Institute in London. Today, I'll talk about some of the work that I have done over the years and show you what my plans are going forward to open my own research lab focused on myelodysplastic syndromes. First, I would like to sincerely thank you all for all the incredible support you have given to us researchers by donating your blood and bone marrow. Without your generous donations, it would not be possible to advance translational research in MDS at all. Your contributions are truly invaluable. So, yeah, thank you. Please continue supporting us so that together we can move this research forward and one day discover a potential curative therapy for you all. So let me begin with giving you a brief introduction to MDS. I know many of you are already familiar with it, so I will keep it short. In MDS, we know that the bone marrow does not make enough healthy blood cells. As a result, blood counts are low, and the blood cells that are produced often look abnormal in shape. This is how MDS has traditionally been diagnosed. In some people, unfortunately, the disease can progress and become more aggressive, for example, acute myeloid leukemia. At the moment, treatment options are very limited, as you know. The only therapy that has potential to cure MDS is stem cell transplant, but most of the patients are not eligible, unfortunately, because of the age and also other health conditions. To better understand MDS, let's start with how our cells are. Every cell has DNA, which acts like an instruction manual, telling the cell what to do. DNA is made up of basically four letters, A, C, G, and T, that are arranged in many different patterns, like words in a notebook, basically. MDS develops when changes occur in this DNA. Sometimes whole section of the DNA, such as a long strand of DNA, are missing or duplicated. For example, we can have chromosome 5 that is entirely deleted in some patients or partially deleted. Other times, smaller changes called mutations happen in the individual's gene, which are like sentences within the DNA instruction book. You may have heard in the clinic, some of you have been told that there are mutations, for example, SF3B1, TET2, DNMT3A. So this is how genetically the MDS is diagnosed. So this is a brief introduction about what we know about MDS. Let's go where it all begins. Where do blood stem cells actually come from? Think of your bone marrow, the soft and spongy part inside your bones, as a busy city that never sleeps. At the heart of this city, we have living blood stem cells that are mothers of all blood cells. They are like master builders with two incredible talents. They can make copies of themselves to keep the supply going. They can also create all different blood cell workers our body needs all the time. From these blood stem cells, we can have other blood cell types. For example, red blood cells. They are like delivery trucks carrying oxygen to every corner of the body. We have white blood cells that are like the defense force, basically fighting the infections like bacteria and viruses. Many of you remember COVID-19 pandemic. They were the frontline soldiers doing all the fighting. Then we have platelets that are almost like a repair crew rushing in to stop the bleeding whenever there is an injury. The scale of this is mind-blowing. Your bone marrow produces more than 220 billion cells every single day. So if we can think about it's almost like a factory that runs around the clock, day and night, 24/7, right? Without ever breaking down. That's how efficient this system is. So, and within this system, the blood stem cells are also surrounding by other cell types in the bone marrow. We can imagine them as city planners and neighbors, helping decide whether a blood stem cell could make more copies of itself, take a rest, or turn into a new cell type. It's like a big family, all working together to keep your blood system running smoothly. Of course, like in any system or a family, things can go wrong. When blood stem cell picks up a mutation or it changes in their DNA, what we call a mutation basically, they can turn into abnormal cells called blasts. These blasts don't work properly, and over time they build up in the bone marrow. As a result, there is a blockage in the blood production line, so the bone marrow can't make enough healthy, mature cells. That's why some patients with MDS develop problems, for example, anemia. There are too few red blood cells, which causes fatigue and weakness, low platelet counts, which makes bleeding harder to stop, or patients bruise easily. On top of this, many blood cells in the MDS also die too early. When they break down, they release debris into the bone marrow. They create stress or what we call inflammation, almost like pollution building up in the city. Normally, we have these macrophages. I like to call them like Hoover cells. They move around and they're supposed to be clearing all this debris. But in MDS, these macrophages do carry mutations, hence, their internal wiring may be broken down. Therefore, they may not know what to do and how to do it properly. Let's look at the MDS in a different context now. Let's look at it as an ecosystem. So we know diversity in nature is not just something nice to look at. It's a basic necessity for life itself. Within this ecosystem, for example, we can think this ecosystem like a forest or a pond, a coral reef, or even your own garden. Every species, big or small, plays a role in this ecosystem. Plants produce oxygen and food, insects help with pollination, birds spread seeds. If we lose too much of one component within this diverse ecosystem, the whole system starts to break down. In the context of oceans, let's say, if the coral reefs die off, the countless fish and species that depends on them will lose their home. So there is an imbalance within this ecosystem. So diversity is not an optional thing. It's what keeps our ecosystem resilient, balanced, and capable of supporting life, including ours. Similarly, in the context of human bone marrow, communication that is taking place between the cells, blood stem cells, and their environment is critical for both the normal functioning of this tissue or a factory and also in the context of tumor or cancer growth. In MDS, broadly, this ecosystem can be divided into three main components. We have the MDS blood cells at the top, then we have the immune system, and also the bone marrow microenvironment. So in the bone marrow microenvironment, as I said, we have 20 different cell types that are supporting the blood stem cells. Within these cells that are supposed to be present in the environment and supporting cells, some of them are over on the left-hand side here, such as mesenchymal stromal cells or MSCs. That's what I will call them after now. We have endothelial cells that make up the blood vessels and others. We now have evidence that these environmental cells may actually play a role in keeping the disease going or even might be playing a role in the establishment of the disease. So in future, I would like my lab to focus on understanding this environment as well as the blood stem cells in MDS, especially the role of the macrophages, what these Hoover cells are doing, and the MSCs. If we can figure out how this neighborhood influences the disease, we might be able to discover new ways to treat MDS. In my future lab, as I said, I want to understand how certain cells in the bone marrow, like the macrophages and the MSCs, might actually be helping MDS stem cells grow and spread at different stages of the disease. At the same time, we also want to understand why the small number of healthy blood stem cells that still remain in the MDS patients are suppressed. Why are they sleeping? What is keeping them from waking up and doing their job? If we figure out how these surrounding cells send signals that protect the MDS blood stem cells, we can look at ways to stop the communication And by breaking this harmful communication in the bone marrow ecosystem, our goal is to stop the MDS blood cells from growing and give healthy blood stem cells that are already present in the MDS bone marrow the chance to wake up and take over again. The big idea is to successfully develop a strategy that one day we will have treatments where a bone marrow transplant is not needed because the patient's own healthy stem cells will be able to grow back once the disease MDS stem cells are gone. Now, I will show you some of the preliminary evidence for each component that I'm proposing to study in future. Let's start with some data to show that the healthy stem cells do exist in the MDS bone marrow. This is what we do in the lab. We take blood stem cells from the patients and we then grow them one by one, spaced apart, so each cell can grow independently. Over about 2 weeks, each cell divides and makes up these beautiful colonies. This way, we can study many cells that have all come from the original cell and have the same DNA. Once we have these colonies, we analyze their DNA for mutations. For example, in one patient, the clinical data had told us that this patient had multiple mutations. You may have heard some of the mutations, for example, SF3B1, JAK2, DNMT3A. So after 2 weeks, we looked at these colonies derived from the individual cells. So in the graph, we show each column represents one colony. So as you can see here, this one whole column is one colony, and we're looking at different mutations in this cell or a colony. If a box is colored, that means the cell has a mutation. If it's white, the mutation is not present at all. So as you can see, some colonies have no mutations at all. In this example, 2 out of 48 colonies had no MDS mutations. We have seen similar results in multiple patients, which show that healthy blood stem cells do exist in MDS bone marrow, and these cells do not have typical MDS mutations. We have seen similar data in patients that are in at a higher stage of the disease or a later stage of the disease. So now we know that healthy stem cells do exist in the bone marrow that have no mutations. Let's look at the environmental cells. Let's focus on these MSCs now. The first question we asked was, how are these cells communicating with each other? What messages are they sending back and forth? To study this in the lab, what we do is we take the blood stem cells and supporting cells from the patients. Uh, in this case, it was MSCs, and grew them together. After 7 days of growing them together, we isolate these cells and we look at their RNA sequencing. It's almost like a fingerprinting that you have on your hands. This fingerprinting enables us to find out what message the cell has received or what it's sending. Uh, and so we know what it's supposed to do or what it's doing. So we had two different conditions here. What we have done is we have taken MDS blood stem cells and grew them together with MDS environment. We also taken MDS blood stem cells and grew them with the environment MSCs from a healthy donor, someone without MDS. We also arranged these cells in two different ways. We kept these two cells next to each other, or we kept them far from each other. So although they are far from each other, but they are still able to send the signals to each other as they are present in the same pot. So you can think about this like a neighborhood in a community. Some neighbors, in this case, it will be the MSCs, may encourage bad behavior in the resident MDS blood stem cells, while as the healthy environment might encourage good behavior. And we can understand how the MDS stem cells are changing the environment, if at all. By testing these different combinations, we can see what are the surrounding neighborhood doing and what blood stem cell do when they come in contact with the healthy neighborhood. So after doing the RNA sequencing or fingerprinting, here you are looking at the map of how the environment is doing now when they were cultured with the MDS blood cells. So basically what messages they have been receiving after getting in touch with the MDS stem cells. Let's begin with two examples. On the left-hand side is the fingerprint of the healthy environment, completely healthy, no MDS at all. And on the right-hand side, you can see the fingerprint of an MDS environment. This is MDS blood stem cells that were growing together with the MDS environment. So now what happens if I will take the MDS environment, MSCs, and group them with MDS stem cells? You can see clearly that it has changed. The healthy environment cells started to look more like an MDS environment. This tells us that MDS blood stem cells can change the behavior of the surrounding cells. We also did the opposite experiment by putting MDS environment cells with healthy blood stem cells. In this case, MDS environment started to resemble more like an healthy environment. This shows that if we block the communication from the MDS stem cells to the environment, we can actually reprogram the environment. From our previous experiments that I'm not showing here, which is already published, we have also learned that MDS blood stem cells are heavily dependent on their environment to survive. So if we remove the environment completely in the lab, the MDS stem cells will die. This raises an important question. How do these cells talk to each other? What chit chat is taking place in there? Given that our data suggested that there is a communication between the cells in the bone marrow, so we wanted to understand what is involved in this interaction or chit chat. We look at something called receptor ligand interactions. Think of it like two groups of workers in a factory. You have these ligands or the workers present on one cell and the receptors or different type of workers present on another cell. When a message is passed successfully from one worker to another, the cell can coordinate and respond. Think it's almost like in a factory, blood stem cells are the main workers, and the MSC is the environment helpers within the environment, keeping everything running smoothly. And in MDS, the helpers in the environment can be rewired by the diseased blood stem cells, which sends messages that makes the MSC support the MDS cells instead of healthy cells. So our goal is to find out which receiving workers on the MSCs are listening to these harmful messages. And if we can block this communication, can we kill the MDS stem cells? So here you can see at the bottom, we have the MDS stem cell that was in direct contact with the environmental MSCs. So what's happening on the MDS stem cells? Apologies, as it's a busy slide. Here you can see the names of various workers. On the left hand, you can see the proteins. These ones over here, proteins are workers from the MDS blood stem cell talking to the workers from the supporting MSCs in the environment. The darker the color, the stronger the conversation, the louder the chatter. As you can think about almost listening like a walkie-talkie. Some channels are quiet, while as others are buzzing nonstop with messages. That's exactly what is happening here. And when we take a look at, let's focus on one communication signal over here. Imagine a worker from gene A is getting the signals from gene B or a worker B from the hematopoietic cells. So whenever we discover a data that is new and hasn't been published before, we need to be really careful and check if the data is real. We need to confirm that what we are seeing in the lab conditions is not just random noise. So we need to look in this context if this worker, i.e. worker A, is really present on the environmental cells MSCs. Here you're looking at a piece of bone that was donated by an MDS patient. So thank you again, shows how precious and useful these samples are. We use a special technique in the lab. What this does is it allows us to let the cells glow in different colors, almost like giving them colored flashlights so we can see who is who. So in this context, you have the green lights, meaning it's coming from an MDS blood stem cell. The red light means it's an environmental cell or MSC. If I will now zoom in one of these parts, you will notice it also, this red cell has a blue light shining inside it. That means the blue light is coming from the gene A that we identified in the previous slide. So our data is real, which is really, really helpful. So for the first time, we can actually see this worker A is present on the environmental cell of the MDS patients. So what happens if we now remove this worker A from the environmental cell? What effect does that have on the MDS blood stem cells and healthy blood stem cells? To test that, what we have done is we deleted the worker gene A or worker A from the environmental cells. And we then put MDS blood stem cells and healthy blood cells, stem cells on top of these environmental cells. These cells were grown for 4 weeks. Just to make a point, the healthy stem cells that we used aren't coming from healthy donor. It's not coming from MDS patient. So now, if we look at the graph and focus on with what is happening with the healthy blood stem cells, in this graph, you have the black and pink bars that show how well the blood stem cells are doing when worker A is present, which is the black bar, or when it's absent, which is the pink bar. For the healthy stem cells, removing worker A has had small impact. Blood cells are still growing fine. But for the MDS stem cells, it was a different story. Without worker A, as you can see in from the pink bar, they struggled and eventually died. You can think of this almost like cutting off a troublemaker's main supply line. The healthy workers in the factory keeps running fine, but the MDS troublemakers can't survive without it. This shows that MDS blood cells are highly dependent on the worker A and the message that it sends. We are now confirming this result in a larger group of patients and also studying other workers that we have identified to see what roles they play in this communication network. But this data is quite encouraging. So I have shown you the data that demonstrates that we can potentially cut the signal between the MDS stem cells and its environment, and it looks quite encouraging in terms of killing the MDS stem cells. This research needs to be done at a bigger scale to confirm this. So now let's talk about the other cell type, which is our macrophages or Hoover cells, and try to see if they are functioning at all in MDS. This is what we have done. We took some monocytes. These are immature cells and encouraged them to develop into macrophages. This is the similar process that normally takes place in our blood and bone marrow. Then we gave these macrophages some external signals like instructions to see how they will respond. We tested this with the cells from both the healthy blood and MDS patients. In healthy donors, as you can see here, the macrophages, these Hoover cells that act like cleaners normally look round, but when we gave them a signal, they changed their shape from being round to more like a fried egg shape at the bottom here, showing they were responding and adapting to the new instructions they received from us. In contrast, the MDS macrophages behave very differently. Even after receiving the same signals, most of them stayed around, as you can see here, and didn't change at all. It's like having workers on the road who refuse to follow the directions. While the healthy workers adapt and get the job done, the MDS workers just sit and do nothing. This is a preliminary data that demonstrate that these macrophages in MDS are not functioning as we expect them to do. More research needs to be done to understand what and why these cells are behaving abnormally. So to conclude, I hope I have shown you some data that shows the MDS blood stem cells do exist in the MDS bone marrow. We need to look at more patients to gather this evidence. We have also shown you that MDS, our Hoovers, seem to be not functioning or responding to the external messages that they receive. This set of results gives us a very, very strong foundation to dig deeper into the bone marrow environment and build more evidence along these lines. And yeah, more work needs to be done. Looking ahead, what I want to do is I want my lab to focus on studying bone marrow environment in MDS and also look at as a whole, as a bigger picture, not only focus on the MDS stem cells. We know that MDS stem cells rely heavily on the surrounding environment, as you can hear. And we also have evidence now that the healthy blood stem cells do exist in the marrow, but they are just sleeping. Our goal is to see if we can block the communication between the MDS stem cells and their surrounding environment. So what we can do is we can let the disease cells die. That will create the space and signals for the healthy stem cells to wake up and start growing. The ultimate idea is that over time, the bone marrow could be repopulated by patients' own healthy stem cells, potentially eliminating the need for the bone marrow transplant that we all know most majority of the patients are not eligible for transplants anyway. So how are we going to do it? We want to generate a Google map of the bone marrow cells, see how these cells are talking to each other, and develop new strategies to stop this crosstalk or the interaction that is taking place between the cells. Using this data, we also want to develop new mouse models potentially that will emulate MDS as a disease. This gives additional tool, not in the context of MDS, but also other diseases. All this data can be used to understand what is happening in the context of other myeloid cancers, such as acute myeloid leukemia as well. It will be a really good stepping stone for us to understand these diseases. With that, I would sincerely thank everyone. All the research we do in the lab is truly a team effort, and it would not be possible without contributions of so many people. I am deeply grateful to my mentor, Dominique Bonnet, for giving me the support and freedom to explore my own ideas. I also want to thank our funders and, most importantly, our patients. Without your generosity and participation, none of this research could happen at all. I would love to continue this journey together with you guys and hopefully one day find cure for MDS.